Well, first of all, thanks very much to the organizers, to Ida and John and Brad for organizing this symposium and for inviting me to present work from my lab in this wonderful setting at Dartmouth College. I'm really very grateful to be here. Now, in my talk, I'm focusing on ERP correlates of face recognition, so that aspect of neurophysiology, in contrast to the previous talk, a rather sort of macroscopic view on neurophysiology is the topic of my talk. But I would still like to start with a functional model, and this is from a paper that Mike Burton and I published about 10 years ago. But really, it's only just an updated version of the famous Bruce and Young model, which tells us that recognizing somebody by their face is a rather complex multi-component process. Oh, is that switched off? Right. Oh. Can you hear me now? Better? Okay, thanks. Okay, so recognizing someone by their face is a rather complex multi-component process, and actually I just want to make a very general point here. In the old days, when I started event-related brain potentials research, it was common to think of an ERP component as reflecting a specific mental subroutine. And with that in mind, you would think that in order to understand the neurophysiological correlates of such a complex multi-component functional system, our general strategy should actually be to study a whole variety of ERP components and other neurophysiological phenomena, depending on which specific process we are interested in. And in my understanding, this has been a point in many subsequent models, also in the famous model by Jim Hexby and Ida Corbini on the neural system of face perception. But it turns out that this sort of multi-component view doesn't seem to be what most in the ERP research community seem to have been doing, and you do not need to do a Web of Science analysis to realize that ERP research in face perception has seen an enormous focus on the N170 component. But unfortunately, this was at the expense of other important ERP correlates of face perception. It is as if we ignored the functional models a bit and believed that we can understand such a complex multi-component system of face recognition by studying just one mental subroutine and its ERP correlates. And that's an unfortunate situation, I think. But fortunately for us and also for our brains, there is much, much more to face recognition over and above the processes that are reflected in the N170 or in the N170 time range. And this multi-component perspective is the point of my talk. So as a start, you might ask why there has been this strong focus on the N170. And I could say, well, I don't know really. But that's not quite true. Here are a few factors that might have contributed. So perhaps the N170 is simply so prominent a response that it is hard to overlook. And of course, a big factor will have been the excellent work that has been done by researchers like Shlomo Bentin, Bruno Rosion, Martin Eimer, and many others. But part of the N170 success story is probably because it represents a potentially face-specific brain response. And of course, the question of whether faces are perceived using some sort of specialized neural hardware has concerned many researchers, and long before the N170 was even in the picture. Now, personally, I do not think that specificity is remotely among the most important questions to ask about face perception. But even if you believe that specificity is a crucial issue, the bare facts around it are rather sobering, I think. So the specificity debate has actually absorbed many, many productive researchers. Huge amounts of research money have gone into the debate, and literally hundreds of papers have been published around it. But I think it's fair to say that many more than 40 years after the issue has been first raised by Robert Yin, there is still no consensus on the issue. So I will happily leave that debate to others. I'm simply interested in all ERP correlates that help us to understand the various sub-processes in face recognition. And this again is, of course, the multi-component view. So today I will focus on 4 components. That you see here, the N170, the P200, the N250, and the N400. and I have selected those 4 components, not because they represent an exhaustive list, but rather because research now suggests a relatively clear and consistent functional interpretation, at least to me. And I have to apologize in advance if this talk will be a little bit like a sampler, but the field has really become so big now that any conventional time slot isn't nearly enough to present you the full picture, so I had to be a little bit selective. So first, there's some context to understand some of the experiments that are going to follow. We often borrow the priming paradigm from experimental psychology to investigate ERP correlates of face recognition. So here a participant has to recognize a target face, often saying whether that face is familiar or unfamiliar by button press, let's say. And the point here is that this target face can be preceded by a prime stimulus, which can be the same face in the same image or the same face in a different image. So in this case, we have either an image-specific or a more abstractive variant of repetition priming. But in other experiments, the target face can actually be preceded by the face or also the name of a related or an associated person. And the idea is that different kinds of priming facilitate face recognition at different stages in processing. And this is what you can actually show very nicely with ERPs. So this is actually from one of our first studies on face priming, which we published quite a while back, as you can see. And these are ERPs to familiar faces that had either been primed by repetition, that's the solid lines, or same faces when preceded by another face, the dotted line. And the blue arrow points to the N170, and this component is not sensitive to the repetition of familiar faces. I'll show you this in more detail and different studies later. In red, you see the N250R. R stands for repetition, so this is an ERP modulation for repeated faces with a latency of about 200 to 350 milliseconds and with a ventral temporal distribution. And this is a response that's typically larger over the right hemisphere, as you can see here. And in green, you see the N400 modulation with a latency of about 300 to 600 milliseconds and a more central to parietal distribution. And the N400 is also moderated by repetition priming, as you can see here. But as we will see, and this is in contrast to the N250R, the N400 is also moderated by associative or semantic priming. Now, almost at the same time, we published this Shlomo Bentin, who, as you probably know, we sadly lost in a tragic accident in summer 2012. Shlomo published this paper, which you of course all know because it really changed our field of research, and it described in a large number of systematic experiments the N170 ERP response. And that paper was probably one single reason for why face researchers had focused so much on the N170. It's an excellent paper, and I just want to point out to you the fact that Shlomo's interpretation of the N170 was extremely far-sighted right from the start. And I think 2 points deserve particular mention here, so I'm trying to highlight this here. So there's this one point where he says, N170 may reflect the operation of a neural mechanism tuned to detect as opposed to identify human faces, similar to the structural encoder suggested by Bruce and Young, 1986. And the second point I want to highlight is where he says, however, the differential sensitivity of N170 to eyes in isolation suggests that N170 may reflect the activation of an eye-sensitive region of cortex. And I think both these points of Shlomo at the time have stood the test of time remarkably well. Now, I've organized the rest of the talk into 4 theses. The first thesis is that the N170 reflects the detection of a face-like first-order configuration and perhaps early aspects of what Bruce and Young called structural encoding. The N170 is no correlate of individual face recognition. Well, firstly, the N170 has turned out to be completely unresponsive to the familiarity of a face, which was shown by many studies, so these are just a few here. But also, secondly, the N170 is largely insensitive to individual phase repetitions, although I will mention a possible exception in a minute. But there's actually another point and a factor to which the N170 is massively sensitive, and surprisingly, this wasn't known until a few years ago. The N170 is strongly suppressed whenever a phase is preceded by another phase, regardless of the identity of that phase, and that's an effect that I call categorical phase adaptation, or categorical repetition effect, it's also sometimes called. So here's some evidence. This is a paper on repetition priming in which a familiar phase was either preceded by a different phase, that's the black traces, or it was preceded by the same phase in the same image, that's the red traces, or the celebrity's face was preceded by a different image of the same person, that's the blue trace. And these are ERPs to the target phases, and you can see no repetition effects whatsoever for the N170. But what you do get is a very prominent N250R repetition effect, and this is much larger for same image repetitions, but it's still present for different image repetitions, and the topography is the same as far as we can determine from scalp-recorded EEG. And actually, this attenuation for different image repetitions is very similar to what you get in behavioral studies of phase priming. Now, this is from a more recent paper by Stefan Schweinberger, in which it is claimed, if you look at the title of that paper, that the N170 is sensitive to phase repetitions, even though this is only the case in the right hemisphere. And this claim actually refers to this rather small effect, which was significant at extremely short stimulus onset asynchrony, so when there is an extremely short time between the prime and the target phase. Now, I'm not saying at all that this finding shouldn't be taken serious. I find it a very interesting finding, but I still found it surprising that this was the main focus of this paper, and in any case, the much more prominent repetition effects that they also observed in the N250 component were hardly even mentioned in the discussion. So perhaps this illustrates a little bit what I mean when I say there has perhaps been a degree of overfocus on the N170 in the field. So despite this interesting study, I think it's fair to say on balance, if you look at all the evidence, that the N170 is certainly not much influenced by individual phase repetitions. And more evidence comes from a study that was published in 2011 by Shlomo Bentin's group. And these are ERPs to phases that were preceded either by the same phase, what they call same image phase adaptation, or by a different phase, that's what they call categorical phase adaptation, or the phase was preceded by another object category. They used pictures of Easter eggs for some reason. Anyway, but what you can see is that the N170 is reduced whenever another phase preceded the target phase. I'm not sure whether you can see this, but you can see the N170 here. So it's reduced whenever another phase preceded the target phase, but at the same time, it's clear that the N170 is completely sensitive to whether or not this preceding phase is the same as the target phase. And I've just picked up these older data because they remind us that the response around 170 milliseconds doesn't even require a real phase at all, and we knew this for a long time. So this is from Jeffrey's studies, and he published in the late 80s and early 1990s. He recorded the vertex positive potential, but it's now thought that this is functionally equivalent to the N170. He just had a different and smaller set of electrodes and a different reference. But this positivity here at 170 milliseconds is essentially functionally equivalent to the N170. And that response at 170 milliseconds is sensitive to completely artificial facial configurations as long as they are arranged such that they are perceived as face-like. So this captures a bit what I mean when I say that the N170 reflects the detection of a face-like configuration. And even completely different stimuli might share some aspects of a face-like configuration, and it is for good reasons that many face researchers, including myself, now have come to love car fronts as a control, really full frontal pictures of cars as a control stimulus to compare with real faces. So car fronts share with faces a number of things. They share with faces symmetry, if you compare them with front faces, of course. Symmetry, the existence of single features in the midline, the existence of lateral symmetric features such as the headlights or the mirrors. And also they share with faces the fact that the general spatial layout of these features, the so-called first-order configuration, is the same across virtually all exemplars. Now, Bruno Rosion in 2000, and perhaps even more clearly our study in 2004, showed that car fronts can elicit substantial N170 responses that can be hardly distinguished from the face N170. And you see this when you compare the red and the black traces here. I'm sorry, I can't really point very well because I don't have the feedback from the pointer, but I hope I point in the right direction. So basically, if you compare these traces, and you can also compare the topographies of the N170 for faces on the left-hand side and for car fronts on the right-hand side, and they are virtually indistinguishable. So the conclusion here could be that the N170 is sensitive to the detection of a face-like configuration. Now, before you think that I might claim that our brain treats car fronts just like faces in the N170 time range, let me say that I'm certainly not in that camp, and the story, as usual, is a little bit more complicated. And in brief, there's good evidence that the eyes play a really special role in face perception, as for instance, Roxane Itier's work from Canada has shown us for quite some time. But as far as the N170 is concerned, the special role of eyes only becomes evident when you look at the data in the context of effects of inversion and feature removal. I can't explain you the theory behind it now, but in a paper that was just published last year, Nadine Claude actually could very clearly show a differential influence of the eye region in upright and inverted faces. And this is the effect I'm referring to here on the top. But by contrast, there was no evidence for a similar interactive encoding of eye-like features, in this case the headlights, and the configuration for non-face objects for the car fronts, even when they possess a similar sort of first-order configuration as faces. Okay, with that I stop on the N170 and I move on to my second thesis, which is that the subsequent P200, occipitotemporal P200 component, is sensitive to the encoding of second-order spatial configurations. Now, the term second-order spatial configurations refers to the relative metric distances between features of faces, and put more specifically, my thesis is that the P200 is actually sensitive to the deviation of a face's second-order spatial configuration from that of a prototypical face, in other words, to our individual perceptual norm. Now, you may be familiar with Tim Valentine's face space concept, and the idea here is that each face is coded as a location in multidimensional space, and the dimensions of this multidimensional face space refer to those aspects in which faces differ from one another. And the origin of face space then corresponds to the average of all faces represented, the average of all faces with which we have perceptual experience. Now, the P200 has actually been related to the typicality of a face in a couple of earlier studies. And actually, it has also been related to the population expertise an observer has with a group of faces. And I'm showing you a little bit of evidence on this. But the most important aspect of data for my thesis here comes from a number of recent experiments that looked at the effects of spatial caricaturing on the P200. And I'll show you some of the data here. So here's some evidence from the other race effect first. So this is basically population expertise. And we found that the P200 on top here is quite selectively reduced for other race faces, at least in German participants, without any contact to Asian faces. That's what you see in the top graph. But in another group of German participants who had documented and long-lasting personal contact to Asian people, this P200 effect was attenuated or absent for the very same faces. So this is an effect of expertise. And a similar effect was also seen in our first study on an own age bias in face recognition for other age phases. So this is from a study Holger Wiese published in 2008. And the P200, as you can see here, was reduced for old faces in young participants. It wasn't reduced in older participants. And at the same time, it was shown that old faces were difficult to memorize for young participants. Again, the full story is a little bit more complicated, but I just wanted to mention this as an initial evidence. Now, the most direct evidence that the P200 reflects the encoding of second-order spatial configurations comes from a number of recent papers that Jürgen Kaufmann published, in which we use spatial caricaturing. So here you see an original face on the left, plus two versions of spatial caricaturing, which just means that every spatial difference between an individual face and a prototypical average face has been exaggerated to different degrees. And I hope you can see that texture and reflectance actually was unchanged by this manipulation. So it's a purely spatial manipulation. Now, the first thing that Jürgen showed is that spatial caricaturing improves face learning of initially novel faces. And this is what you can see here in accuracies and reaction times. And this benefit of spatial caricaturing is probably because it makes a face more distinctive. At the same time, spatial caricaturing also elicits a very systematic reduction of the P200. The right hemisphere electrodes are on the right-hand side. So what you can see here, the P200 is large for veridical faces. If you look at the PO10 electrode, where it's clearest, it's reduced a little bit for moderate degrees of spatial caricaturing, and there's a massive reduction for strong degrees of spatial caricaturing. There's again much more to this story. I just would like to mention one point briefly. Our data suggest across a number of experiments that spatial caricaturing is helpful to actually learn to encode novel unfamiliar faces. But at the same time, spatial caricaturing doesn't seem to be helpful for the recognition of already familiar faces. And this points to important differences in the representation, in the mental representation of familiar and unfamiliar faces, which I think are a little bit underappreciated in the literature potentially, but this would be a different talk. I'm just saying that there's a lot of behavioral work by the groups around Vicky Bruce and Mike Burton and Peter Hancock who actually study this difference in representation between familiar and unfamiliar faces, and our findings are entirely consistent with their analysis. Now, the P200 modulation, to come back to that, doesn't seem to reflect mere spatial distortion relative to a veridical face. Rather, it really seems to reflect the level of deviation from the typical second-order spatial configuration of an average face. And this conclusion is derived from this paper by Claudia Schulz, in which the effects of spatial caricaturing and spatial anti-caricaturing were compared. That is, by anti-caricaturing, you make a face spatially less distinct, so you move the spatial configuration towards the average face. And actually, Claudia found that the effects of caricaturing and spatial anti-caricaturing on the P200 were exactly opposite. So on the right-hand side, you see again that caricaturing reduces the P200 amplitude. This is what we already knew. Anti-caricaturing increases the P200 amplitude. And again, those effects were particularly prominent at right hemisphere occipitotemporal electrode sites. So for now, my short take-home message is that the P200 relates in a systematic manner to the encoding of second-order spatial configurations. My third thesis is that the N250 or N250R component indexes individual face recognition of familiar faces. Now, firstly, the N250R is clearly smaller for unfamiliar face repetitions. It's very prominent for familiar face repetitions. But let me go straight to the evidence. You've already seen that this component responds to face repetitions, but I briefly want to mention the issue of source localization of the N250R repetition effect, that is the difference between repeated and unrepeated faces. And the point here is that we consistently found a source of the N250R, which is shown here in red, in the fusiform cortex. This is found across a number of experiments in EEG. We also found it in MEG. But of course, these are scalp-recorded data with all their limitations. But for actually blue is the location we find for the N170. But we also did an fMRI study together with Rick Hansen in London. And you see the results of this fMRI study, companion study on the right. And you see an intriguing similarity in the sense that familiar face repetitions clearly modulate the activity in bilateral fusiform regions, a bit more strongly so in the right hemisphere. And by the way, I should perhaps mention that the N250R doesn't like car fronts at all. And in that respect, it's perhaps more face sensitive than the N170. So what you see on the top left is actually the difference between repeated and unrepeated stimuli for different categories. And you see this large N250R response for human faces. You basically see no modulation for cars. And if you look at the topographies, you see that there is a strong right hemispheric ventral temporal effect for face repetitions, which actually, if you move to primate face stimuli, this is actually reduced, but it seems to sit in the same place. For inverted faces, there's almost no repetition effect. There's a statistical trend here, which is suggestive. And for cars, there's absolutely no N250R repetition effect. More recent study by Markus Neumann and Tariq Mohammed was in a slightly different context in which attention to the primes was manipulated by a perceptual load task. So participants attended, if you look at the left column, they attended to either high load or low load letter strings, and they decided whether or not they contained the letter X or N. And these strings were superimposed on briefly presented faces or houses or hands, which were used as primes, unattended primes here. And the ERPs you see on the right-hand side are from the target stimuli. And these target stimuli could be repetitions or non-repetitions of these unattended distractor primes. And you see the typical large right occipitotemporal N250R repetition effect for faces, but you also see no N250R modulation for repeated houses and also none for repeated hands. This study by Markus Bindemann shows that the N250R is entirely preserved when primes are geometrically stretched relative to target faces. And this is, of course, based on the pioneering work by Graham Hole, who had published a paper in 2002 in Perception, a spectacular paper rather, where he showed that you can actually change the aspect ratio, the linear stretching of celebrities' faces by a dramatic amount, up to 300%, with virtually no cost to identification performance. That's almost hard to believe, but the data are really quite good, and you should look at the paper if you haven't seen it yet. And we've just used this as a tool to see whether, if we manipulate the primes in that manner, whether or not the N250R repetition effect is affected. And the study shows actually that the N250R repetition effect completely survives this kind of stretching, which suggests that the N250R indexes face recognition. At least it doesn't simply index the visual overlap between two successive facial images. So much for familiar face repetitions, but what about unfamiliar face learning? Well, the figure on the left is from a paper by Jim Tanaka's group, and here participants learned a particular target face, which is termed Joe here, and the ERP during learning was compared with that to other non-learned phases, and also to an overlearned phase, which in this study was always the subject's own phase. And as you can see here, perhaps the familiar own phase, which is shown in black here, always elicited a large N250 response. The learned phase, which is shown here, let me see, in blue, the learned phase initially behaved like an unfamiliar phase, but after learning, in the second half of the experiment, the learned phase elicited an N250 that was as large as the N250 elicited by the familiar phase. And those findings were confirmed and extended by Jürgen Kaufmann in a collaborative study with Mike Burton, which showed that the N250 gradually increases over the course of phase learning, even when different images of the same phase are shown in different learning blocks, and we thought this was an important extension. There's also evidence that the N250 indexes covert face recognition of familiar faces in cases of developmental prosopagnosia. And this study is from the London group around Martin Eimer and also Brad Duchesne, and it reports data from no less than 12 individuals with developmental prosopagnosia. And in 6 of those individuals, these are the data from them, non-recognized famous faces triggered an occipitotemporal N250 component. So this is shown here in red, and note that negativity is plotted up in this figure, which makes the waveform slightly unusual to look at, for me at least. But this is an N250, and these authors also reported no indication of later ERP effects that would have suggested late semantic stages of face identity processing. And this is, of course, consistent with a lack of explicit recognition of familiar faces in these individuals. So these findings are also in line with my thesis that the N250 reflects the activation of identity-specific visual memory traces for faces. But they also suggest that it is a disconnection between those visual memory traces and later semantic memory for people that has prevented conscious recognition to occur in these individuals. So this is all good and nice. The interest in the N250 has grown quite a lot in the past few years, and there are now many people from other labs like Martin Eimer and Jim Tanaka and others who do research, and their results are perfectly consistent with our interpretation of the N250. But you may actually have seen some of our own very new papers, particularly the excellent work by Holger Wiese, who's here in this room today. And you might wonder how they fit with this story. So before you are puzzled, I just want to mention a little caveat. I actually refer to the occipitotemporal N250 or N250R as an ERP difference between repeated and unrepeated phases, or between learned and novel phases. And it's in this situation where the N250 is really a consistent correlate of the transient activation of representations for the individual recognition of faces. But we need to be aware that many simultaneous neural computations can affect the amplitude of occipitotemporal face-sensitive ERPs. So take the N170 for an example. The N170 is small or absent in some people with prosopagnosia, and then it's interpreted as indicating deficient perceptual encoding of faces in these people. But at the same time, the N170 is particularly large to inverted as opposed to upright faces, and in this situation, this is also interpreted as an indicator of deficient perceptual encoding. So this is an apparent paradox. Sometimes a large N170 relates to good performance, sometimes it relates to poor performance. And very similarly, although I'm sticking to my interpretation of the N250, we have to be careful not to overgeneralize the N250 interpretation when it comes to different groups of faces. For instance, own race or own group or other group faces, attractive or unattractive faces, and so on. And I'm just mentioning this because I know that many people in this room here are interested by those categorical differences. And actually, these group differences between faces can also exhibit modulations of occipitotemporal processes in the N250 time range, but these modulations may reflect a different process, maybe something like emotional tagging, a process that can actually interfere with memory. So take this new paper from Holger Wiese on the own race bias as an example. So this is a fully crossed design in which we tested 20 Asian and 20 Caucasian participants in an episodic memory paradigm with own race and other race phases. And in behavior, both these groups exhibited a substantial own race bias. They were better to recognize faces from their own ethnicity. Now look at the differences in the N250 time range to differences according to race. So Asian observers show more posterior ERP negativity. That's what I've highlighted in blue here, to Caucasian than to Asian faces. Whereas Caucasian observers, that's what's highlighted in red, show exactly the opposite finding. So basically, there is an own race bias in the sense that, and of course, the other race phases were always the ones that were less well recognized, and they elicit larger negativity here. And intriguingly, there is a substantial correlation between this individual neuronal race effect and the behavioral own race bias that you see on the bottom left. Now, how do we interpret this? Well, we believe, but this is still, novel data. We believe that in this situation, the effect might be related to an EPN component, an early posterior negativity that has been described for emotionally salient stimuli, for instance, in Harald Schupp work. So we think what could be going on here is that this larger negativity might reflect the tagging of other race phases as emotionally salient, and this could actually interfere with the recognition of these phases. Still preliminary, but actually a very similar point can be made from another new paper Holger published earlier this year, in which we studied the effects of facial attractiveness on recognition memory. And I think I have to skip a detailed description of this study in the interest of time. But if you're interested in the real effects of attractiveness on memory when controlling for facial distinctiveness, which hasn't been done in a lot of the previous research, then perhaps you want to look at this paper. Now, just very briefly, the paper shows that attractive faces are actually less well recognized than unattractive faces, and at the same time, attractive faces also induce a relative bias to believe that a face has been encountered before. So these are both behavioral effects that you can see. And attractive faces also elicit a larger early posterior negativity in the N250 time range. And this amplitude difference also correlates on an individual basis with the individual difference in D prime, that is in the memory disadvantage of attractive faces. Okay, with that I come to my last thesis and to the N400 component, and the N400 modulation in the context of person recognition studies reflects domain-independent person recognition and the access to personally semantic information, and not, it doesn't reflect face recognition specifically. Now this is from an old study that investigated associative priming in person recognition depending on the stimulus domain of prime and the target. So this is a kind of Gorbachev-Yeltsin type of priming. And the main finding was that associative priming quite selectively modulated the N400, and this modulation was the same for priming within and across stimulus domains. So on the left-hand side you have ERPs to target faces, which were either primed by prime faces within domain or primed by celebrities' names cross-domain, and you see the same N400 modulation. On the right-hand side you see entirely the same experiment, except that the targets were now written names presented on the monitor, and you can see essentially the same N400 modulation. It's also independent of whether you prime within or across a domain. And I think this is quite remarkable because, of course, the overall ERPs for target faces and names are very, very different. They're just very different visual stimuli, but still the modulation caused by associative semantic priming is entirely comparable. And this is why I'm saying that the N400 appears to reflect a late post-perceptual or semantic processing stages in person recognition rather than in face recognition. But what are the mechanisms of associative or semantic priming, and how exactly is semantic priming for people organized? Well, semantic priming could be organized along categories such as British people, American people, politicians, actors, and so on, or it could be organized along associations that reflect the real life experience that we have, that some people tend to co-occur together. Now, this is not easy to study because although you will find many people that are categorically related but unassociated, it's a bit more difficult the other way around. And some of you will perhaps know the influential interactive activation and competition model by Mike Burton. And my friend Mike Burton always claimed that semantic priming is mediated purely via activations that utilize those categorical relationships as you can see them here. But I'm sure most of you will not know that Mike and I had really heated discussions about this over more than a decade. And in all this time, I didn't believe a word of it. I was absolutely convinced that pure categorical priming in person recognition in the absence of association simply didn't exist. Okay, so I didn't believe this stuff. So what's the evidence from current ERP work? Well, actually, Holger Wiese tested this by investigating priming for faces that were preceded by masked prime names, either at very short or at long stimulus onset asynchronies. And the primes were either associated to target persons or they shared the same occupational category, but they were unassociated. And intriguingly, at short SOAs, both associative and categorical priming initially elicited identical N400 effects. I'm just highlighting in green here the size of the categorical priming effect. So this is indeed evidence for the priming mechanisms that is proposed by Burton's IAC model. But also this effect of categorical priming dissipates very quickly and has almost gone away by 1 second of SOA, whereas the associative priming effect is actually further boosted, such that associations seem to play a dominant role for semantic priming at more typical longer SOAs. So that's not a bad outcome perhaps for both Mike and me, because in this case we were both right perhaps, a little bit. Or depending on your perspective, you could say we were both a little bit wrong depending on your threshold. So in summary, what do ERPs tell us about face recognition? In a nutshell, they tell us a lot. At an early stage of the detection of a face-like first-order configuration, we see a consistent reflection in the N170 component. Now, slightly later, the occipitotemporal P200 seems to reflect the encoding of second-order spatial configurations of faces quite specifically. The subsequent N250 or N250R indicates the activation of facial representations for individual face recognition. And finally, the N400 is sensitive to the post-perceptual access to semantic representations about people and not just about faces. And of course, you note that there are quite a few gray areas left in this schedule, and for some of them, intense research is going on as we speak. But for now, I hope that I have convinced you of my one general point, which actually is that in order to understand the neural implementation of our sophisticated abilities in face recognition and face perception, we cannot just continue to focus on isolated ERP components or on particular time ranges or on particular isolated brain locations for neural responses to faces. But instead, we need to study the specific way in which a whole variety of ERP components and other neurophysiological phenomena reflect different sub-processes in face and in person perception. Actually, when I wrote the abstract for this talk, I thought I might have a little bit more time to tell you about our work in person perception more generally. We run a large DFG-funded research unit on person perception in JENA, and there we are interested in how humans perceive and identify people from several senses. And we study not only visual but also auditory perception of human voices. And like you will, I'm sure, hear in more detail by Pascal Belin in the next talk. We also think that there are a lot of parallels between faces and voices in the sense that they can trigger social cognitions, but this is really something that I'm looking forward to Pascal's talk, and I've just listed a few of the sort of more overview papers that could be an entry if you are interested more in the work that we've been doing recently, also on the issue of audiovisual face-voice integration and on the issue of speaker perception. So that's the end. I would like to thank all my colleagues, my postdocs and my PhD students, without whom this research wouldn't have been possible. Thanks also to our funding bodies, and in particular to the DFG for rather generous funding of our research unit. And I want to particularly thank Holger Wiese, who is here today, but who will not be in JENA for very much longer because he joins Durham University as a senior lecturer very shortly. But thanks, Holger, for the great research and the fun we had while sharing quite a few productive years in the JENA lab. And obviously, thank you for your attention.
The talk is open for discussion.
So Stefan, the idea that there are neural mechanisms that detect eyes makes a lot of sense to me. But I've always been confused by the idea that the eyes, that response of those neurons is inhibited by the face, given that the face probably, co-occurs pretty well with the eyes. Yeah. So what do you make of that?
Well, I'm not Roxane Itier, and so it's not my model, and I think this is the model we are talking about, right? And I think in a way, in the description, it seems to be a bit underspecified to me. But at the same time, a lot of the empirical data, also the ones that we find when we work with similar experimental, manipulations as she does, sort of eye removal, presentation of single eye regions, and so on. I mean, the results are fairly consistent with that interpretation, and also we are not. I think we should become a bit better in source imaging and source localization as well. This is not our particular strength, but I think the data we have collected on this so far also tend to be consistent with the idea that the N170 actually that you record at the scalp is governed by at least two major sort of generators, one of which is really sort of eye sensitive and the other is really face sensitive, and they actually interact with one another. But there's actually a lot of sort of complex and interesting issues around the eye. Is like there is probably one process of eye detection, and there's a secondary process of actually determining social attention or eye gaze direction, and these are not reflected in the N170 time range to the same degree. As far as I understand the data, eye detection is really happening in that period, but sort of determination of eye gaze direction happens at a slightly more advanced stage in processing and slightly more anterior areas in the STS, as also the work by Andy Calder has shown.
So since you mentioned source localizations, in one of your slides you were pointing out the fusiform localization. I forgot if it was the P200 or the N250.
Yeah.
And the more posterior location of the N170, was that also in the fusiform?
No, in that study it was actually in the lateral occipital temporal cortex. So that's more sort of consistent with OFA, I would say.
I see.
But the limitation of that particular paper was that we had only been, our data were well modeled by just one single source, but as we just discussed, I think actually this is only an oversimplification. That's probably the dominant source that you get for the N170, because this OFA location, it might also have to do with the general generator orientation. And actually scalp-recorded ERPs are extremely sensitive not just to generator localization, but also to generator orientation. And that might be why it's actually easier to pick up this kind of OFA or lateral occipitotemporal source for the N170 in EEG. In MEG, it might be a little bit different.
But so despite these caveats, could you say like where you think the P200, N1, N250, and the N400 might be localized relative to each other?
Yeah, I mean, that's a good question, and actually it lines up nicely with what you said about, the degree of abstraction of the representation actually increasing as you move sort of from more posterior to more anterior sides. And I think in general our data are entirely consistent with that. We have also, done source localizations on the N400 modulation, and if we do that, we get a source which is more anterior in ventral temporal lobe, so it's sort of anterior hippocampus and very anterior fusiform. But we've never published this because I've always felt uneasy with localizing, such late ERP responses where probably you would just have a multitude of generators acting in combination. And if we have only scalp-recorded ERP data without much, converging evidence, say from fMRI, I would have been, or I was too careful to actually publish this. But in general, I think our results are quite compatible with this sort of posterior to anterior gradient.
So you showed that N250 is responsive to facial attractiveness. Do you know if it's responsive to other types of aesthetic beauty?
To other types of aesthetic stimuli?
Aesthetic beauty, let's say bodies or architecture or something like that.
Well, I don't know that from my own research, but actually we've interpreted those findings more in the context of this EPN, of this early posterior negativity modulation. And this is actually work coming more from the affective processing literature. So Harald Schupp has originally and others described this component as a response to emotional stimuli. And so I would think that it actually would respond to a number of sort of visual dimensions that are capable of eliciting that type of response. But yeah.
I was wondering what your thoughts are on oscillations which are not maybe stimulus locked, but evoked.
Well, that's a very good point, and actually we want to move more into neural oscillations because we have, but this was not my original area of expertise, so we're just about to publish a study on voice recognition, actually, which is coming out in the Journal of Neuroscience next month, where we try to find correlates of the recognition of individual voices that you either previously learned or that are novel. And this is a situation where we didn't find any good indicators in standard ERP responses, so we just had at least no real correlate in the standard ERP of what we found in the behavior, which was substantial learning of voices even when you use entirely different utterances at the test. And in time frequency analysis, we actually could find, in this case, a suppression of beta band activity in right temporal areas and at around 280 to 370 milliseconds. So that was quite nice. And the general point is, I think you're right, we need to look at sort of time frequency analysis more frequently in the future, and we haven't, we are only just starting to do this, but I'm actually very excited about doing more in this area.
Sorry, maybe just to go back to the previous question, if you can clarify again why you interpret this 250 in terms of affective tagging and not in terms of the familiarity with this particular configuration or knowledge of both the
well, basically, because then yeah, that's a good question. But basically, then we would be faced with the paradox. Then we would be faced with the paradox that actually those stimuli, in this case, the other race phases or the more attractive phases, surprisingly, those stimuli that are actually less well recognized in the test elicit a larger N250 response. But of course, it's a comparison between different categories of stimuli, and not a comparison between, actually the same physical stimulus, depending on whether or not it has been learned or familiarized before, or whether or not it has been primed by repetition.
But attractive faces sometimes are also more prototypical, right? So they are more.
Yes, yeah, that's a good point. So I mean, I could have mentioned this, and you might actually want to look at the paper, because this was the main point in this. Attractive faces are more prototypical, and we assessed actually 2 measures of distinctiveness to see whether distinctiveness is a moderating variable for the memory effects of attractiveness. And what we found is exactly what you say. If we correlate rated attractiveness across something like 600 faces, if we correlate rated attractiveness with ratings for what I call deviation-based distinctiveness, so that the instruction is something like, how much does this face deviate from typical faces you know? Then you get this correlation in the sense that more typical faces or less distinctive faces are also rated more attractive. But intriguingly, if you use another definition of distinctiveness, which is also common in the literature, and that's the face in the crowd measure. So if you ask people, how easy do you think you would recognize this face in a crowd of people? And this is also commonly used as a proxy for distinctiveness. Then we get exactly the reverse correlation. I mean, they're moderate correlations, but they're highly significant. So attractive faces tend to be the ones for which people say, well, I would detect this face in the crowd. So what this suggests to us is actually that there's probably a gap in the literature as well. You can't just use these 2 proxies for distinctiveness to reflect the same thing. They correlate positive with one another, but only by about 0.4 or 0.5 or so. And they correlate in opposite direction with attractiveness. So the way we think about this is that maybe, we think the deviation-based measure for distinctiveness is a better one. And we think that the face in the crowd measure perhaps induces a kind of cognitive bias, in the sense that maybe you say, oh, this face is so beautiful, surely I would detect this in the crowd. We don't know this, but this is our explanation. But